Hello everyone and welcome to this class. In this class we are going to study about the transition of aerobes to anaerobes. So basically how the aerobic bacteria they transform their metabolic structure and their overall condition so that they can survive in anaerobic condition or in lack of oxygen. So let us start with a little explanation here. See, this we have a bacteria here, okay? So this small part is its nucleus and this whole complete cell is this, with this being its cytoplasmic part, okay? So first of all, say this is HIF, HIF1 alpha. So this is HIF1 alpha, HIF1 alpha, okay? So this HIF1 alpha have a proline in it, okay? So under normal condition, when oxygen level is normal inside the bacterium, so what happens is PDH, this prolyl hydroxylase enzyme, this PHD, prolyl hydroxylase enzyme, this comes and this hydroxylate this proline, OH, okay, hydroxylate this proline, and this whole HIF1 alpha complex will get ubiquitinized and proteasomal degradation will be there, okay? Since there is proteasomal degradation, then you can say this HIF1 alpha, it will not function in normal oxygenic condition. But when oxygen level drops, then what happens? Then first of all, this PhD functioning will stop, okay? If this PhD functioning will stop, then there will be no hydroxylation, no ubiquitinization and no proteasomal degradation of HIF alpha 1 will be there, okay? So after this, what happens is this HIF1 alpha will go inside the nucleus where it will meet HIF1 beta, okay? So this HIF1 alpha plus HIF1 beta, they will form a heterodimeric structure, okay? And let us say we have our DNA here, and these are the promoter sites, okay? So there will be a specific sequence here which are HRE sequence. Let me write it here, HRE sequence. Here, suppose this contains the HRE sequence, HRE sequences, okay? So this heterodimer will go and bind themselves to HRE sequence, which are hypoxia response element sequence, and the DNA will start to transcribe in such a way that all those genes which were toward this side, they will transcribe, and these genes will transcribe in such a way that this bacterium will convert itself into anaerobic condition. Now we will study what are the changes that can occur here when it is going to convert itself into anaerobic condition, okay? So first of all, when the oxygen level drops, then you can say the first thing which is going to translate are the GLUT transporters, GLUT1, okay? So these are glucose transporters, okay? So let us say this GLUT1, they are formed here. A lot of GLUT1 will form and they will move to the plasma membrane, and they are a transporter. What they will do, they will bring inside glucose, okay? And apart from this GLUT1, some glycolytic enzymes will also form there, glycolytic enzymes. So glycolytic enzymes will also form, and these glycolytic enzyme will help in glycolysis. means breakdown of this glucose. So our glucose will break down using glycolytic pathway. You know all the pathways like glucose 6 phosphate, fructose 6 phosphate, fructose 1,3 bisphosphate, and then glyceraldehyde phosphate, etc., etc. So at last it will convert to pyruvate. So okay, so glycolytic enzymes will work here. Okay, glycolytic enzymes will work here, so that glucose will get broken into pyruvates. And another kind of enzymes which can work here is, it will get translated, those are lactate dehydrogenases. Okay, lactate dehydrogenases. I'm so sorry, the suddenly light went out. So as I was saying that it will also produce this lactate dehydrogenase enzyme. It will convert this pyruvate into lactate. Now the conversion of pyruvate into lactate is very important because it will help in regeneration of NAD positive. Okay, if this bacterium was following its normal metabolic cycle, then this particular step is not required, because in normal process the NAD will be formed. But since the anaerobic conditions are getting developed here, so you can say some other conditions. So some other conditions are also necessary in order to regenerate this NAD positive. Okay, here because glucose is being converted into lactate here, so you have to know that the ATP production in this anaerobic condition, ATP production will increase, but on the cost of less efficiency of this breaking of this glucose. Okay, so you can say more number of glucose will be broken to generate more number of ATP. This is slightly a more glucose consuming process compared to the aerobic bacterium's metabolism, but it is needed because oxygen is less in this case. Okay, yeah. So let me also complete it that when all these things, okay, yeah, remember these three parts, it is very important. Okay, when all these metabolic changes occurs, then this whole bacterium will start to develop its metabolism in such a way that everything will get adapted to anaerobic condition. Basically these are the points. Basically this is how energy will be generated if oxygen is less there. But what you have to also notice is, what you also have to notice is that while this transition is occurring, there are certain changes that also occur. Like we have gene expression system. So first is this gene expression system. As you can see that this HIF1 with the proline is continuously, it will not degrade here. So it will continuously bind with HIF1 beta in the nucleus, and these genes will be activated. So only those metabolism, okay, second thing is metabolism. Those metabolic processes will change, or you can say metabolic things will occur which are necessary for this anaerobic bacteria to survive. Okay, in this metabolic process, the amino acids will also change a little. So you can say protein and lipid structure will change a little, so that overall morphology of this bacterium will change. Okay. So these are the changes and how this transition from aerobes to anaerobe occur. Thank you for watching my video. I hope you liked it well. Thank you very much and see you soon.